Good morning, sir.
Good morning, madam.
Good morning, sir.
Good morning.
Sir, inge Pandi Kumar.
Good morning, Pandi. Good evening,
Sir.
Now it's the time to introduce our resource person for session 1. I am extremely honored and privileged to introduce Dr. K.P. Ilango sir, Professor, Department of Chemistry, Gandhigram Rural Institute, Gandhigram, Dindigul. He done his MSc and MPhil degree from Annamalai University. He graduated with doctoral degree in coordination and complexes from Gandhigram University. Dr. K.P. Ilango sir is an expert in teaching with 30 years of experience. Sir has published 209 articles in reputed scientific journals. 20 doctoral scholars have graduated under his direct supervision. 7 scholars are currently doing PhD under his supervision. He completed 2 CSIR and 2 UGC and 1 Government of India major research projects. Sir received UGC Mid-Career Award and now he is the Director, University Instrumentation Center. His research interest areas are coordination complexes and optical sensors. Now the session is open on small molecules as anion sensors. Over to Dr. K.P. Ilango, sir.
Thank you, madam, for your nice introduction. Just a minute, I will. Slide is visible?
No, sir.
Oh.
No, sir.
So now it is visible, sir.
Okay, okay. Okay. I will start it. Okay, friends, good morning to all the participants and the organizers. At the outset, I would like to thank the organizers for giving me this opportunity to share some of our research ideas with the young chemists, particularly PG students. Okay. So I think majority of the PG students have enrolled in this faculty, though it is a faculty development program. Many participants are PG students, right? So today I am not going to teach you any new chemistry. This lecture is particularly planned in such a way that it is to motivate young researchers, particularly young staff members, those who are working in colleges, just to motivate. See, everybody can do research. Only thing, you have to read more and more research articles, and you have to think logically. A small amount of thinking is more sufficient to conceive or to design a research problem. Okay. Then another thing is, many of the speakers nowadays will tell that we have to do some more applied research rather than basic research. In my opinion, when applied basic research walks 10,000 steps, then only applied research can walk only one step. So always basic research is very much needed in any field of science or in any field of research. Okay. Don't be afraid of those people. They are, in my opinion, they are all innocent people. Basic research is must. So here I am going to give examples more from our laboratory, what we have done in my laboratory. So I think I can take another nearly an hour to present this. Say, so let us go into the topic, small molecules as anion sensors. Okay. So this gentleman is a saint. Kindly pardon me, இடையில தமிழ்லையும் சொல்றேன், since it is திருக்குறள். Okay, மோப்பக்குள்ளையும் அனிச்சம் முகம் தெரிந்து நோக்கக்குள்ளையும் விருந்து. Say he is a saint, தெய்வ புலவர். Why? Because he has not left anything untouched. அவர் பேசாத விஷயமே கிடையாது, எல்லா விஷயத்தையும் பேசியிருக்காரு. So he is a saint rather than a simple poet. இவ்வளவு பேசின ஒரு சென்சிங் பேசாமல் இருந்திருப்பாரா? Say, in my opinion, this couplet, this Tirukkural speaks about the principle of sensing. See, மோப்பக்குளையும் அனிச்சம். Anisam is a flower, is a thermal sensor. When the heat is radiated on this flower, it charges. Then முகம் தெரிந்து நோக்கக்குள்ளையும் விருந்து. What is this? This is the basic principle of a sensor. See, there are three events. One is the guest analyte. நம்ம வீட்டுக்கு வர்ற ஒரு கெஸ்ட். So whether it is a cation or anion or a small molecule, so first the guest analyte, then the receptor unit. Though யாரு நம்மளை ரிசீவ் பண்ண போறாங்களோ அவங்க, the host, கெஸ்ட்டை ரிசீவ் பண்ண போறவங்க. Then the signaling unit, what actually it signals, and the hostoட face, முகம் தெரிந்து நோக்கக்குள்ளையும் விரிந்து, அந்த ஹோஸ்டோட ஃபேஸ் எப்படி போகுதோ, வாங்க வாங்கன்னு கேட்கறதுக்கு வா அப்படின்னு சொல்றதுக்கும் நிறைய வித்தியாசம் இருக்கும். ஸோ தட் இஸ் தி ஆக்சுவல் ரிசப்ஷன். அந்த ரிசப்ஷனை பொறுத்து அந்த கெஸ்டோட மனசு, அந்த மனசு தான் சிக்னலிங் யூனிட். ஸோ ரிசப்ஷன், தி அனலைட், ரிசப்ஷன் அண்ட் சிக்னலிங், இது மூணுமே இந்த 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 குறளில் இருக்குது, இந்த திருக்குறள், இந்த கப்லெட்டில் இருக்குது அப்படிங்கிறது இட்ஸ் மை humble opinion. Okay. So what are anions? See, we thought that immediately when we said anion in a classroom, immediately chloride, bromide, iodide, fluoride will strike our mind. So we thought that these are all the only anions present in chemistry. We have n number of anions. So anions has a very crucial major role in our daily life. Whether it is a physiological function which takes place in vivo, or it is a environment, or it is an industrial process. Many anions have their significant role to play. Okay. So whenever once we use anions in industry, naturally the effluent will be discharged out. It enters into the environment. There is a very much chance for the anion to enter into our body through water, air, and soil, so that it becomes a harmful pollutant. Sir, is all anions are pollutants? No. See here, I have listed 3 anions: acetate, chloride, and phosphate ions. All these 3 anions are very much needed for our well-being. They have their own significant biological role to play in our body. Say, common building block, acetate ion, digestion of food, chloride ion, and the phosphate ion to give energy, to produce energy. Okay, so they have their own biochemistry in our body. So these anions are very much needed for our well-being. Even if you take whether small amount or a large amount, it doesn't affect the biological system, or it will not act as a pollutant even at a relatively higher concentration, right? Then here I have given a list of other 3 anions: nitrate ion, sulfide bisulfide. Of course, these 2 ions are the derivatives of sulfur dioxide. So commonly they are known as sulfur dioxide pollution. If you say that it is sulfur dioxide pollution, then the hydrated sulfur dioxide, hydrolyzed sulfur dioxide, sulfite and bisulfite ion are the major pollutants. Then cyanide ion. If you look at these anions, they are pollutants even at very low concentration. Nitrate ion, of course, nitrite, nitrate, organic nitrate, inorganic nitrate, all ions. Then if you look at sulfite and bisulfite ion, particularly bisulfite ion is extensively used as a preservative in many foods and beverages, even wines, jams, chips. They contain sufficient amount of bisulfite ion as preservative, since it blocks the oxidation reaction. So when the concentration of the sulfite bisulfite increases, it becomes a pollutant, cell death occurs. The nitrate ion gives a blue baby syndrome, it affects the heart. Then cyanide, as we all know that it is lethal poison. Okay, so here I have not mentioned one more very important ion, namely fluoride ion. I will keep it for next class, namely tomorrow I will speak more about fluoride ion. So anions can be very biologically significant anions. They are non-pollutants, or these anions are common anions which are pollutants. So today's lecture I am going to concentrate more on cyanide ion. Okay, the cyanide ion sensing using very simple molecules. Then when you talk about sensing, detection, recognition, all they have almost, they are synonyms. Suppose if you want to detect nickel ion, immediately DMG will strike our mind. So DMG can be used to detect nickel ion qualitatively in a given solution. The same dimethylglyoxime DMG can be used to estimate nickel ion either through calorimetrically or through gravimetrically present. Quantitative estimation is also possible. So we need to have a similar molecule like DMG for nickel. We need to have a similar molecule which can detect qualitatively and determine quantitatively the anions in particular. So this lecture is designed in such a way to concentrate more on anions, particularly cyanide ion. Then when we talk about cyanide ion, we all know that it is deadly poisonous. Even there is a joke that the taste of cyanide ion is still not reported. So such a fast acting cyanide ion, it causes death immediately. Then we thought that cyanide ion is present only in laboratory. It is not anywhere outside the laboratory. It is not so. It is not correct. So here I am going to give some facts about cyanide ion. See, look at the sources of cyanide ion. Say the cyanide ion is available in nature or it can be man-made. Then there are certain bacteria, fungus and algae, they can produce cyanide ion. So biological system can also produce cyanide ion. Then shockingly, certain food materials, plant materials, they contain cyanide ion as along with the sugar molecule in nature. So food plant materials also contain cyanide ion, but not in ionic form, but in a molecular form, as in the form of an organic cyanide. So many cyanide in soil water came from industry, that is the man-made source. So naturally fungi, algae and bacteria can produce certain food materials, they have cyanide in their as organic cyanide, but many industries, just I have listed some of the industries here, they are using cyanide ion for variety of purposes. So this is about the source. So food, it is shocking that food materials, they contain cyanide ion, right? Then how cyanide enters into our body? By all means, say either through air. There are industries which can emit cyanide containing exhausters. So cyanide, for your information, even cigarette smoke, it contains trace amount of cyanide ion. Inorganic cyanide is present in cigarette smoke. Okay. So once the industries uses huge amount of cyanide for many processes, the discharge, the effluent naturally will contain cyanide. So water is another source. Similarly, soil. So cyanide can enter into our body through many means. If you look at the concentration of cyanide in drinking water, it ranges from 0.001 to 0.011 ppm. Though it is very small, it contains cyanide, right? Then more interesting. So cyanogenic glycosides. As I told you earlier, many food materials, they contain the cyanogenic glycosides, which are basically phytotoxins. Phyto means materials derived from plants, plant kingdom, toxin. So it is toxic. It was reported that nearly 2000 plant species, they contain this particular cyanogenic glycoside. So here I have given some list of materials. The figure, look at the figure. Apple, particularly apple seeds, they contain huge amount of the cyanogenic glycoside. It varies from one variety to another variety. So that is why I have not given any quantitative picture here. Just I have given the figure alone. Then the cassava roots, ஒன்றும் குச்சிக்கிழங்கு மரவள்ளி கிழங்கு அப்படின்னு சொல்லுவாங்க. cassava root, say this is a major source of food in many countries, including India. Even we have chips varieties, even the so-called murukku. Murukku is made up of the cassava powder. Okay, கடையில் கிடைக்கிற சாதாரண சின்ன கடைகளில் பெட்டி ஷாப்ஸில் கிடைக்கிற முறுக்கு is made up of cassava. So then tomato, cherries and almonds. The almond, there are two varieties. One, the picture shows the normal variety. Then there is another variety known as black almond, bitter almond. That bitter almond is it is a food supplement for diabetic patients. It is commercially available, both almond and bitter almond. When compared to almond, bitter almond, badam paruppu, bitter almond contains more amount of the cyanogenic glycosides. So it is very common around us. Say apple seeds, cassava, potato, cherry and almond are commonly used by many people in the world say these are the 5 food materials. They contain relatively higher amount. If you look at some research article, the concentration of cyanogenic glycoside in cassava ranges from 1 milligram per kilogram to 1000 milligram per kilogram, nearly 1 gram per kilogram. So each variety has its own amount of cyanogenic glycosides. Okay. So what are cyanogenic glycosides? So here is the structure. A sugar unit is attached to a carbon attached to cyanide. So it is an organic cyanide, CCN. It is simply an organic cyanide. So this is the structure of the cyanogenic glycoside. Okay. So is it harmful? No. As long as it exists in the form of a CCN bond as an organic cyanide, it is harmless. It can enter into the body, it can be metabolized, it can be digested, and it can be released unaltered without any harmful effect. But during that metabolism, by chance, if basic condition, particularly basic substances, has the chance to interact with the cyanide, this CCN bond can be cleaved under basic condition to release inorganic cyanide, namely cyanide ion, Cn minus. Here it is not exist as Cn minus, it is only CCN, but it can be released in our body so that it becomes a pollutant. So cyanogenic glycosides are the main sources of cyanide in our body. There are many reports. You look at this, the total this work published in Journal of Food Composition and Analysis during 2012, cassava food products in India, in Australia. So this is the basic source which is available. Don't think that cyanide is present only in the laboratory, not else anywhere. It is with us in our day-to-day activity. Some more references. This is this review speaks about the cyanide in all possible edible plants. Then this recent review published in 2017 in Environmental Science Pollution Research speaks about the ill effects and the concentration of cyanide in the environment, particularly soil and water. So this is how cyanide enters into our body. Food materials can also bring cyanide into our system. Right. See why cyanide is such a deadly poisonous substance. Here is the basic mechanism. See, I am not going to talk about the detailed biochemistry. So just look at that. We have a cytochrome oxidase, an enzyme in our body. The main function of this enzyme in electron transport to produce ATP. ATP is the energy source, the compound, an organic compound, which gives energy to our cells for its function. Okay, so cytochrome oxidase through many biological function, it produces ATP. During this synthesis of ATP in our cells, there is a reversible exchange between Fe2 plus ferrous and ferric ion. Ferric is converted to ferrous and ferrous is converted to ferric. Of course, there are some intermediates where we have Fe4 plus also in the biochemistry. Okay, so this is a reversible normal cell respiration which is happening in our cells when cytochrome oxidase produces ATP through electron transport. When cyanide enters into our body, it coordinates with the Fe3 plus ion leading to cell death. So this is the basic mechanism. So it hits the cytochrome oxidase and it coordinates. We know that cyanide ion is a very strong ligand. Being an inorganic teacher, if you go through the coordination chemistry chapter, there is a term spectrochemical series. If you look at the position of cyanide ion in the spectrochemical series, along with the carbon monoxide, cyanide ion occupies the last position. So such a strong ligand, it coordinates with the Fe3 plus and thereby causes cell death. That is why cyanide is a poisonous substance. Okay. but this occurs at a very fast rate. Okay. Then what are all the symptoms due to cyanide poisoning? Say here it is classified into two. One is mild poisoning. Say when we take cyanogenic glycosides through metabolism, it can release very small amount of cyanide in our body. If the concentration is relatively higher, it is mild poisoning, leads to headache, anxiety, vomiting, and suffocation. So these are all the basic symptoms of mild poisoning of cyanide in our body. When the concentration increases, what happens? To moderate to severe poisoning, it leads to cardiac arrest. So a rapid loss of consciousness, weakness or absence of breathing, and cardiac arrest, naturally death. Okay, so moderate to, then for information, according to World Health Organization, the maximum concentration of cyanide is in drinking water. The permissible limit, the allowed limit is just 1.9 micromolar, 1.9 into 10 power minus 6 molar. 1.9 micromolar is the maximum permissible limit. So even in the case of mild poisoning, once the patient recovers, even after survival, it causes brain injuries due to its toxic effect. Okay, due to lack of oxygen in the blood, right? So such a poisonous substance cyanide is, right? Is there any remedy? Antidote is available for cyanide. In the case of mild poisoning, it is possible to remove cyanide from our body. But if the concentration is higher, it is not possible. So here again, I have not given any quantitative estimation. This is because the moderate and severe or low poisoning depends upon the body weight. It is actually the milligram per kilogram of the body weight. So I have not given any quantitative picture. Just I have classified into mild poisoning and moderate to severe poisoning, right? In the case of moderate poisoning, it is possible to remove cyanide. How? Just by injecting or by taking metallic ions like cobalt 2 plus, iron 3 plus, so that it can coordinate with the free Cn minus and it can be expelled from the body. So metallic ions, by taking large amount of metallic ions, but if you take large amount of Fe3 plus and cobalt 2 plus, it has its own ill effect in the body, but no other go. To remove cyanide ion, this is one method of treatment. Then the naturally available vitamin B2 hydroxycobalamin can be used as an antidote. What actually happens? The cobalt present in the vitamin B12 can coordinate with the cyanide ion so that it can be removed, right? Then enzymatic conversions. There are certain enzymes which are present in our body, has the capacity to convert minimum amount of cyanide into a non-toxic thiocyanate ion, but it cannot be done in a large manner. It is very small amount of cyanide can be converted. Then once if the cyanide containing air is inhaled, amyl nitrate can be given to release the cyanide, to remove the cyanide by inhalation. So these are all the possible antidotes to cyanide in the case of mild poisoning. If it is moderate to severe, the patient, the individual cannot be saved. Only death is the ultimate effect, right? So what are all the main approaches for anion sensing in general, particularly for cyanide ion? I have given this list for cyanide ion, but any anion can be sensed, can be detected in any one of this method. So the first one is hydrogen bonding approach. So as we all know that anions possesses a negative charge, it can interact with hydrogen bond donor molecules, the organic molecules which contains H plus or H delta plus. So acidic hydrogen, it can make hydrogen bond with the molecule. So if you look at this picture, so the pink colored ball is the signaling unit, and the green colored C is the receptor unit. Once the anion or cation, any analyte binds to the receptor unit, the signaling unit gives a signal that can be viewed either through naked eye or by means of any instrument. Say the signal can be a change in color, change in fluorescence, change in electrochemical behavior, change in change in even mass, any property, any observable property. So if it is color change, we can visualize using our simple naked eye in daylight. If it is fluorescent change, that can also be viewed using naked eye under the UV light, but other changes can be measured only using instruments. So always the optical detection, either colorimetric or fluorimetric, is the simplest. It doesn't need any sophisticated instrument to record the change. So this is hydrogen bonding approach. I can say around 75% of the papers published so far for anion sensing uses this methodology, namely hydrogen bonding approach. Definitely 75% of the papers uses this approach, but this approach has its own disadvantage. I will tell you later. Okay, then displacement approach. This is somewhat closer to reaction based approach. Say here, look at that pink colored ball in the B, second in the picture, the second one B. This is a fluorescent or a colored substance. When the analyte enters, it displaces the signaling unit, so it glows, so it gives color change or it fluorescent change. Then the third one is a reaction based approach. Say this is very specific or selective approach for sensing anions. Say whenever a chemical compound, a receptor, a chemosensor is used by reaction based approach, that sensor is known as chemodosimeters. So chemodosimeters are chemical sensors which senses anion through permanent chemical change. Okay, so these are all the three main approaches. There are even complex formation, ligand substitution. There are many approaches are there, but basically these are the three main approaches to sense anions. Okay, then see what makes anion sensing difficult in our aqueous system in water. Say for practical application, we need to have detect and determine a particular analyte in aqueous solution. Say for example, as I told you earlier, take DMG. DMG can be used in aqueous solution to detect nickel 2 plus ion. Similarly, DMG can be used to determine the amount of nickel ion in aqueous solution. Okay, so here when in the case of anions, it is very difficult to detect them or to determine them in aqueous solution. What makes this difficult? The high hydration energy of this anion makes it very difficult. So as I told you earlier, among the three major approaches which are reported so far, hydrogen bonding constitute 3/4. So here the high hydration energy, look at sulfide ion, fluoride ion, they have extremely high energy. That means they love water molecules like anything. So this is the amount of energy in kilojoule per mole when one mole of the particular anion undergoes hydration. So they are fond of water molecule. Whenever water molecule is present, they undergo hydration so quickly with the release of huge amount of energy. So your sensor molecule, when it operates through hydrogen bond, it has to overcome the hydrogen bonding between the anion and water molecule, particularly in aqueous solution. Then here in the bottom, I have given a picture. Look at, say, this is experimentally and theoretically proved that in cyanide ion, both carbon atom and nitrogen atom can form hydrogen bond with the water molecule. So this is against our basic theory of hydrogen bond. So during our undergraduate or even during the school days, we told that more highly electronegative atoms can form hydrogen bond with water molecule. Here carbon is not at all an electronegative atom, but it is also capable of forming hydrogen bond with water molecules. So it was theoretically and experimentally proved that each cyanide ion will be surrounded by 7 water molecules. So that minus 67 kilojoule per mole of amount of energy is released. So in the case of cyanide, the energy is relatively less. If you look at acetate, fluoride and sulfite ions, it is very difficult since they have very high hydration energy. So this high hydration energy of anions prevent them from detection in aqueous solution. But for any practical application, we need to have a sensor molecule that can detect or determine these ions in aqueous solution. Okay, so with this basic facts about cyanide ion, I will move to some of the works that are carried out in our laboratory for sensing of cyanide ion. I am not going to cover any other ion today, just cyanide ion alone. So particularly we are interested in recognizing cyanide ion in food materials, cassava powder, almond and apple seeds and so on. Okay, so how to design a molecule? As I told you earlier, it is very easy. Only thing is you have to think logically. See, here I have given a famous reaction of cyanide ion. Benzaldehyde reacts with cyanide ion to form a compound known as cyanohydrin. Cyanohydrin is an active intermediate in many synthesis in organic chemistry. So from cyanohydrin we can synthesize many variety of organic compounds. So such a significant intermediate the cyanohydrin is. So if you look at the history of cyanohydrin, it was reported by Winkler in 1832. Imagine, the cyanohydrin formation reaction was discovered in 1832, but in 1966 the same cyanohydrin formation followed by its reaction with some other molecule is used as a sensor to detect cyanide ion, particularly in basic medium. So cyanohydrin uses its reducing property, it reduces some other molecule so that it gives a color change and so on. So further chemical reaction, it was published in Analytical Chemistry during 1966. So many of you may not born during 1966. Okay, so nearly 55 years has gone after the report of first cyanide sensing in basic medium. Till today, we are struggling to find out a molecule, a sensor molecule, to detect cyanide ion in aqueous solution, even after 55 years. See, the basic chemistry behind this. During the formation of the cyanohydrin, you have an anionic intermediate. Whenever in the para position you have para nitro and para cyano substituent, a strong electron withdrawing substituent, this anionic intermediate can be stabilized through resonance. This is the structure I have given. So when you have an electron withdrawing substituent in the para position, the intermediate, anionic intermediate can be stabilized so that it undergoes hydrolysis to form cyanohydrin. This is the basic chemistry behind this. See here in this, during this period between 1832 and 1966, many aldehydes and many substituted benzaldehydes with the different substituents, nearly 40 substituents has been tested. Similarly, many anions, not only cyanide ion, many anions, the reaction between anion and aldehyde, benzaldehyde, the carbonyl group of aldehyde has also been tested. Then it was found that only cyanide ion can react with the benzaldehyde. That too, when you have a strong electron withdrawing nitro or cyano substituent in the para position. So here the chemistry is, we need to have an electron withdrawing substituent in the para position to the carbonyl group of benzaldehyde. So during the past 10 years in our laboratory, we are working with quinones. We know a little bit chemistry of quinone, but we all know that quinone is a strong electron withdrawing substituents. So we thought that we not introduce the electron withdrawing substituents in the para position to the formyl group. So we have prepared this compound. It is easy. Take dibromo naphthoquinone, then the Suzuki coupling with the boronic acid, formyl boronic acid to yield this product. Okay. So it is a very simple molecule. The chemistry is, you have a formyl group which can react with the cyanide ion in basic medium according to the earlier reports. Then in the para position, you have an electron withdrawing substituents. Here we have selected quinone. Then another advantage in quinone is that it can act as a signaling unit. It can, through charge transfer, it can vary its color behavior also. Okay. Then similar anionic intermediate, it can also be stabilized through resonance structure as in the previous case, as in the case of para-cyano and para-nitro substituent. Interestingly, we found that the cyanohydrin formation takes place not only in basic medium, in all acidic, neutral and basic medium. Look at the color change. It gives an intense dark green, but only thing is, since it is a reaction based one, it takes some time. The color intensity goes on, increases with the time. Nearly it takes 20 minutes for the reaction to complete. Then we have measured the red constant under pseudo first order condition. These are all the pseudo first order red constant. If you look at the pseudo first order red constant, naturally in the basic substance, basic medium, it is relatively fast, but it is comparable in all the three mediums. So this is one of the advantage of this particular sensor molecule. It can sense cyanide ion, but it is not instantaneous. It takes some time. It requires at least 20 minutes for the reaction to complete to give such an intense green color. This green is known as bottle green, such a intense green color. But from pH 3 to, we have done this experiment with variety of pH from acidic to alkaline medium. It gives the same intense color. So just how to design a molecule. Say this is one example. Okay, go through the literature, understand the chemistry, then think logically. You can design a particular molecule for that purpose, right? Then the same molecule, just extend it. See, like the formyl group present in this receptor molecule, we can undergo reaction with the cyanide ion. The imine part, the CIF base, the CH double bond N, this reaction site, this particular functional group can also undergo similar chemical nucleophilic addition reaction with the cyanide ion. So just extend it. What happens? So instead of formyl, we have introduced just a couple with an amine to form an imine, a CIF base. See, same molecule as in the case of previous one. Any amine for that matter, it can be coupled. So here we have selected a thiazolamine. This is because whether the nitrogen and oxygen can coordinate, just to check it whether it can coordinate with any other cation also. Okay. So interestingly, here the color of the solution of the receptor after upon addition of cyanide ion changes to blue. Okay, so it acts as a colorimetric sensor for cyanide ion in DMF solution. But the main disadvantage is, to dissolve this molecule, we need around 80% DMF. Only 20% water can be accommodated in the reaction medium. Okay. So the fluorescence of the receptor also changes by the addition of cyanide ion. Initially it is yellow in color under UV light, then after the addition of cyanide ion it gives a blue fluorescent. So colorimetrically it gives a color change. Similarly fluorimetrically it also gives a change. So how to test the mechanism, how to investigate the mechanism. So this signal around 9.2 corresponds to the imine proton, CHN proton, CH proton. So after the addition, it moves to aliphatic region, namely around 5. So this indicates that nucleophilic addition of cyanide occurs at the carbon atom of the imine group, CH double bond N, the imine group. Then the fluorescence, turn on fluorescence, the blue fluorescence is due to excimer formation. So excimer just is a union of two molecules. It can be intermolecular or intramolecular. Just theoretically, we have shown that the increase in fluorescence is due to excimer formation. Okay. Then the main part is, look at that. We have taken cassava powder, kuchikalang mavu, then the potato, particularly our sputtered potato, then the almond. The same fluorescence we are able, just it is a qualitative test. So what we have done is, we have hydrolyzed the cyanogenic glycoside under basic condition. Say at pH 9 to 10, you stir the aqueous solution, aqueous suspension of this cassava powder for overnight for about 6 to 7 hours. Then filter it, neutralize the solution again, then add the receptor. It gives nearly same signal, same fluorescence. Okay. This is this photograph was taken under UV light, same fluorescence. Fine. So this is our main aim. We have designed many molecules to detect cyanide ion, particularly in food materials. Then this is another one. See, this part, diformyl para-tertiary butyl, this is the fluorescent core which is commercially available. Just make an imine. Earlier we had quinone here as a chromogenic unit. Here we have taken a fluorophore. Actually this fluorophore shows green fluorescent in the free molecule. So we have made some imines with 2-aminothiophene. Now it is gives an yellow fluorescent. So here look at the different bottles, containers upon addition of different anions, chloride, bromide, bisulfate, sulfide, cyanide, and so on. Okay. So only after the addition of cyanide ion, the yellow fluorescence changes to green fluorescence. And the basic mechanism as proved by NMR is that the deprotonation of OH takes place. So this is an example of interaction through hydrogen bond. So initially the cyanide ion makes a hydrogen bond with the proton of OH group and it abstracts the proton, leaving an anion. So this anion gives an green fluorescent. Okay. So here the fluorescence spectra shows that with increase in concentration, the peak increases. So the color change was show that it is highly selective towards cyanide ion. Similarly, the fluorescent spectrum also confirms the high selectivity of the receptor towards cyanide ion. All other ions exhibits very marginal change in the fluorescence of the receptor, but only cyanide ion gives a significant change. So NMR titration, you find that the proton around 13.4 or something near to 13.5 disappears. So say on progressive addition of 0.5 equivalent, 1 equivalent and 2 equivalent and so on. So deprotonation mechanism. Here again we are able to visualize the same fluorescent change in cassava powder and potato, but of course it gives a different color with almond. So same fluorescent change, it can be used to detect cyanide present in these food materials. The yellow fluorescent changes to green fluorescent as in solution. This is in solution phase. This is in the extract of the food materials. Okay. Say the same molecule, the same fluorophore, diformyl fluorophore, another imine. So here you have the formyl group, phenolic OH group, imine nitrogen, sulfur, nitrogen. We have variety of coordinating sites, binding sites in the molecule. So we have taken this, we have designed this molecule with an aim that either cyanide can interact with the formyl group, or it can interact with the OH, or it can interact with the imine, or it can also coordinate with any of the cation. That is why we have designed this molecule. Okay, we have variety of reaction sites for cyanide to interact. So initially it shows a red fluorescence. After cyanide ion addition, it gives an yellow fluorescence. So interestingly, here the reaction, the sensing can be done with 80% aqueous solution, aqueous methanol solution. So 80% water, it can accommodate 80% water molecule. Okay. So the yellow fluorescence is turned to the red fluorescence turned to yellow fluorescence. The actual mechanism is deprotonation of the OH as well as nucleophilic addition of cyanide to the imine carbon atom takes place. This was inferred through NMR titration experiments. Okay, we can follow the OH proton as well as the imine proton, CH proton, by adding incremental amount of cyanide to the receptor molecule. Here again, if you look at the fluorescent spectrum, cassava powder, almond and potato shows excellent increase in fluorescence. So we can detect these ions present in the food materials. Okay. Interestingly, this substance can also gives excellent color change in the solid state also. So in the solid state, there is no water molecule, there is no hydration, so cyanide detection can be very easy. So the first one, R, is the receptor. Under normal light, it appears pale green. Under UV light, it is shows an yellow fluorescent. Then after addition of 0.25 equivalent of cyanide, it turns to red. Then 1 equivalent, it turns to brown, a yellowish brown substance. Say this red color is due to what? So initially, first the deprotonation of the phenolic OH takes place, followed by addition. So this has been proved by NMR, by solid state UV, by solid state fluorescence. So this is the fluorescence spectra recorded in the solid state. So first initially the receptor has a very weak fluorescence. Then after addition of 0.25 equivalent, it gives a red fluorescent. Then followed by after addition of 1 equivalent, it gives an yellow fluorescent as the final one. So we are able to visualize the intermediate also. We are able to, so since it is reaction based, it takes some time, so that we are able to take the photograph of the substance. So in solid state, without any hindrance, cyanide reacts and gives a fluorescent change. Okay. A similar molecule we have reported. So this is benzoquinone. An imidazole was constructed on both the sides. This is first and second imidazole, which has the NH group. Through the hydrogen bonding interaction with the NH group, this particular receptor shows a beautiful color change in solid state, in aqueous solution, as well as in gaseous state. It can also senses gaseous cyanide ion. So in solid state, you see that an yellow color powder turns to beautiful pink colored powder. So without any hindrance. In solid, just add potassium cyanide and grind it. That's all. It gives a color, observable color change. Similarly, in 90% aqueous solution, an yellow solution turns to red, pinkish red solution. Similarly, gaseous cyanide. But when we use gaseous cyanide, you should be doubly careful. It has to be done with fume cupboard, with the quenchers and all, which would make sure that only trace amount of cyanide gas is available. It is very easy. Take DDQ and add any acid, it can release cyanide gas, but it has to be done with high precautions. Okay, don't try this one. Right. See, this is another interesting molecule. So before going into the details of this molecule, see, look at that. How we got that idea? How we can design the molecule? See, here I have given the structure number 1. I have given a diquinone. This diquinone was reported during early 60s as a dye, as an organic dye molecule. So a diquinone with extended conjugation can be used as a dye molecule. There are reviews where similar type of molecules has been reviewed. So this is a well-known structure in organic dye chemistry. See, look at the lambda max of this substance. It is 1335 nanometer. It is intensely colored substance. Then the structure 2 in the bottom. See, this is nothing but indigo dye. So here again you have a quinone with diquinone with the conjugation. Then two NH moieties are there. If the NH is deprotonated, it becomes N minus. So that after deprotonation, the absorption is redshifted. So the indigo dye gives coloration after deprotonation through this mechanism. So we need to have a diquinone with extended conjugation. By some means, if you are able to deprotonate the NH group, definitely it is going to give a color change. Okay, look at this molecule. What we have done is diquinone with extended conjugation. So in the quinone gadayala complete alternate single bond, double bond will be there. So extended conjugation. Then there are two NH, two imidazole NH. If at all any anion binds to the NH through hydrogen bonding, here through NMR spectrum it is proved that cyanide selectively interacts with the NH group and deprotonate, it becomes N minus, so that it shows an intense green color. A brown colored compound turns to green, that too with an absorption maximum at 913 nanometer. So lower energy, higher wavelength, lower energy absorption or emission is always required for biological applications. Okay, so how we are designing a molecule? It is very easy. Just go through the literature, understand the chemistry behind that. Then since we are working with the quinones, we have already prepared some imidazoles using quinones. Here we have prepared a diimidazole with benzoquinone. So similar molecule we have prepared. Pyridine dicarboxyaldehyde with the diaminone after quinone gives the molecule. So initially theoretical studies proved that the molecule is non-planar. After deprotonation it becomes paka planar. So any planar molecule will have intense intramolecular charge transfer. That means there is a transfer of charge from N minus to quinone, so that it gives an intense color. Even in test paper, you look at that, it gives beautiful green colored after deprotonation by cyanide ion. So this system can be done in 80% aqueous DMSO solution. It can accommodate 80% water molecule. It is highly selective towards cyanide ion, right? So the same fluorophore, diformyl phenol fluorophore with the tetrabutyl group in the para position. So here what we have done is we want to study the effect of substituents R. we have taken nitro, an electron withdrawing substituent, unsubstituted, untested butyl, an electron releasing substituent. So depending upon the nature of the substituents, it gives different emission in the presence of cyanide. So red emission, intense green emission, and a weak green emission. So we are able to discuss the effect of substituent here. Okay. We found that, so this is the result. Number one, electron withdrawing substituent. So whenever you have an electron withdrawing nitro substituent here, it can polarize the imine bond so that the carbon atom becomes C delta plus, so cyanide addition can occur more easily. So the binding constant and the rate constant is relatively higher in the case of nitro substituent. The opposite is true in the case of electron releasing substituent. So the polarization, since the tertiary butyl is electron releasing, the polarization is weak. Naturally, the positive charge on the carbon atom is relatively less, so addition is sluggish. Cyanide, it can change, it gives color change. So the mechanism is deprotonation of the phenolic OH followed by nucleophilic addition to the imine bond. So just how we have designed molecule, same fluorophore, just we are altering basic chemistry on that, right? So this is another fluorophore, the bis-thiophenol aldehyde. Make an imine. So look at that. After addition of cyanide, so there is no OH to deprotonate. The only possibility is that cyanide has to undergo nucleophilic addition to the imine carbon atom. It gives a color change from yellow to red and an fluorescent enhancement. A weak blue fluorescent becomes an intense fluorescent after the addition of cyanide ion. Okay, take a fluorophore, make an imine. That's all. So, greenish blue fluorescent becomes intense blue fluorescent. Interestingly, we have made another similar molecule with the same fluorophore. Again, we are going to use the fluorine chemistry here. So since quinone is strongly withdrawing, like nitro substituted benzene, the quinone can polarize the CN bond, the imine bond strongly, so that addition can occur easily. So in the previous case, here we have a nitro substituted, an electron withdrawing substituent to polarize the imine bond. Here we have another electron withdrawing substituent in the form of a quinone. So due to reaction with the cyanide, it gives a green fluorescent. So a non-fluorescent molecule becomes a green fluorescent. Here again, cassava powder and almond is able to give same green fluorescent in the presence of the receptor. Okay. Okay, this is another interesting same fluorophore bis-thiophenol with another amine to make an imine. Interestingly, cyanide undergoes nucleophilic addition, whereas hydrogen sulfide cleaves the imine bond. So it gives the same blue fluorescent, but through different mechanisms. One through nucleophilic addition and another one through cleavage of the imine bond to give back the starting material, namely carbonyl and amine. So just keep the fluorophore constant and vary the substituent depending upon the nature of the substituent, nature of the amine group, it works differently. So these two mechanisms have been proved through NMR titration experiment. We found that cyanide has a stronger binding constant than the hydrogen sulfide. So likewise, I think it's my time, or can I go for another 5 minutes? So same fluorophore molecule with a different amine. In solid state also we are able to visualize, this is solid UV and solid fluorescent spectrum. We are able to visualize the same color change from red to blue. Okay. Then this one is a commercially available substance. We have not made any synthesis. This is a commercially available substance which gives a green fluorescence in the presence of cyanide. Then the practical application. So again a quinone chemistry. So here we have proved the resonance effect through single crystal structures. So I will stop at this stage. So these are all some of the molecules. Just I will rush it up. Reported from our laboratory. So here in this case, even the food extracts gives colorimetric change, not only fluorescent change, colorimetric change. Okay, to conclude, see so far huge amount of research papers has been published for the detection of cyanide ion, but still we are struggling to have a substance which works in 100% water, which can be used to detect cyanide ion. So this is because of the high heat of hydration energy and polarity of water molecule. So to conclude, still lot to be done to find out a molecule which sends cyanide ion in 100% aqueous solution that can be used as a test in the laboratory for cyanide ion. So do some magic, find a molecule to test cyanide ion or any anion for that matter that can be used in aqueous solution. Okay. At this juncture, I would like to thank my guide, Professor G. Karthikeyan. No more now. Actually, I did my PhD in coordination compounds, but he only motivated me to do research in charge transfer complexes. So we came to charge transfer complex only through we came to the designing of anion receptors using the charge transfer complex mechanism. So all these compounds involves charge transfer. So I would like to thank my professor, my guide, my guru, my mentor.
Thank you, sir. Thank you for your vibrant speech on sensors. Participant, kindly ask any queries or put in the chat box.
Sir, can you see the questions, sir? Some questions are posted in the chat box, sir.
Ilango sir, I have one question. Am I
Sir,
audible, sir?
kindly unmute, sir. Ilango, sir, you are muted, sir.
Yes, yes, yes. Sir, you are audible, please.
Sir, give some details about heat of hydrations. In one slide, you have removed some cyanide ion,
Yeah.
like heat of hydration energy. In the case of cyanide, it was 67 kilocalorie per mole, and in the case of fluoride, 505.
Yeah. Mm.
It is not possible to remove fluoride in this strategy?
So it is not removal, it is only to detect sensing alone. So
Okay.
it is possible, but when compared to cyanide, since fluoride has a higher hydration energy, it is difficult to do sensing of that ions in aqueous solution. That is one. There are reports. Tomorrow I will show you some examples what we have done with the fluoride ion. Okay. So tomorrow entirely like today's cyanide, tomorrow I am going to talk about the sensing of fluoride ions. It is possible, but once the hydration energy becomes higher and higher, it loves water molecule naturally. It is difficult to sense through hydrogen bond, but still there are plenty of works available, plenty of reports are available where the sensor molecule works through hydrogen bond. It is possible. It doesn't mean that it is not possible, it is difficult, that's all.
Sir, in that slide, you have also shown that OH bonding is surrounded by some CN cyanide ions.
Yeah.
How is it possible to it will make some bond with carbon and hydrogen also? In the case of nitrogen, it is possible.
You mean to say that whether the cyanide can interact with carbon or nitrogen?
The both items, nitrogen atoms can be possible, sir, because due to charge or electronegativity effect. But is it possible to carbon will form bond with hydrogen?
Carbon, which carbon atom? You mean the cyanide carbon?
Yeah.
Yeah. There are, that's why I have shown that. Recently there is a paper in physical chemistry, chemical physics. They have proved that even carbon atom can make hydrogen bond with the water molecule, the hydrogen present in the water molecule. The cyanide carbon can make hydrogen bond with hydrogen atom present in water molecule, but it is against our basic chemistry. We know that only highly electronegative atom can make hydrogen bond, but they have proved that.
No, sir. Actually, the carbon which is sp hybridized, it has some electronegativity, so it can make hydrogen bond, weak hydrogen bond, but it can make.
Yes, but it was proved only recently. There is a extensive theoretical work has been done on that particular system. It was I have forgotten to include the reference. It is published in PCCP. Okay.
Okay. Thank you, Ilango sir.
Okay, thank you.
Sir, it is possible to detect cyanide minus ion in our body by this sensor?
There are works related to imaging of cyanide in cells, but only in vitro.
Okay.
Only in vitro studies are available.
Sir, what should be the minimum concentration of cyanide ion in this sensor? What should be the minimum concentration of cyanide ion?
It varies from system to system. If you look at the
average minimum concentration,
minimum concentration to detect to work. See, in the case of calorimetric sensors, you need to have a relatively higher concentration, say in the range of 10 power minus 4 or 10 power minus 5 molar to see the color change.
10 to the power 4?
Minus 4 or minus 5 molar.
Minus molar. Okay.
But in the case of fluids, even at lower concentration, we can see the color change.
Okay. Sir, is it possible to detect cyanide minus ion?
Cyanide minus ion?
Any DMS or any other solvent? Only aqua solution. Is it possible?
No.
Is there any sensor?
It is very easy to detect cyanide ion in organic media.
But
DMSO, but
my question is it possible in aqua solution? Only aqua solution.
there are reports. Only thing is your system, your sensor should be soluble in aqueous solution. Since majority of the compounds are organic compounds, their solubility in aqueous solution, even we have tried one system, just by adding few drops of DMSO, we can dissolve the particular receptor in water. But it requires, since they are all organic compounds, it requires some water molecule. But there are sensors. By introducing some polar groups, we can make these sensors soluble in aqueous, pure water, neat water, so that we can sense. There are reports.
Okay, okay.
Hello, sir.
Thank you, sir.
What they will do is they will introduce some carboxylic acid group or sulfonic acid group to make it soluble in water.
Okay, okay. Thank you, sir.
Hello,
Yeah,
sir. Am I audible?
yeah, please, sir.
Sir, how to remove cyanide ion from food or water, or how to change
See,
poisonous to the
actually it is not.
helpful species.
See, our aim is to first detect the cyanide present in the. Then we have to think of determination. It is not removal. It is only a qualitative test to test cyanide ion present in the food material. It is well known. Already plenty of reports are there. But how our system, the molecule developed by us, works with the food material, that is the main aim. Like cell imaging, we have tried with some food material. It is not removal. It is only detection. It's a recognition, just to test. So, any other question?
Sir, from water, how to remove it?
So, adsorption is the best technique, is the cheapest technique. There are many reports to remove anions and cations by adsorption process. Even in our department, Professor Meenakshi is working on that. They have developed some many polymeric composite materials to remove fluoride ion. They have not tried any cyanide ion, fluoride ion, chromium 6 ion, some metal ions. They are doing wonderful work. They have made some excellent contribution in the removal of fluoride ion in particular.
यहाँ तो 1.5 गुंठा जगह देखा है बंगले ले रहा
Thank you.
है क्या नहीं और दिया इससे का को भाड़ा आएगा रे
Okay, thank you, friends.
मेंटेनेंस
We have to stick on to the time. So once again, I thank the authorities of NGM College Pulachi, particularly Pumudi Madam, for giving me this opportunity to share some of our research idea with the friends, chemistry friends. Thank you, ma'am. Thank you.
काम नहीं कर करो क्या करें ऐसा करते मेरे ये सुनो अपने को फेंकी हुई दुकान की
Thank you, sir. Dear participants, kindly switch on your video for photo.
2025-26 में ना यूनिवर्सिटी की बहुत जगह निकलेंगी तब ट्राई करें मुंबई की ना मुंबई की 15 जगह है केमिस्ट्री बेकार धंधा नहीं रहता बेकार धंधा नहीं रहता क्वालिटी और जो बच्चे भी रहते हैं ना
What is going on?
इधर के बच्चे आते नहीं कॉलेज सबसे बड़ी दुर्गति है कॉलेज मेरे को अच्छा ही नहीं लगता कॉलेज मेरे को पूरा माइल्ड कर दिया कॉलेज था ना इतना स्ट्रिक्ट बंदा था इधर आके मेरे को पूरा बिल्ली बना दिया चलता है इधर कोई कुछ को कुछ नहीं बोलता है आने का तो मेरा क्या कहना है वो अगर नहीं आता है ना should be punished. Otherwise, you will take action and I'd cancel his admission. क्यों उसको फ्री में डिग्री दे रहे हो तुम?
Participants, kindly mute yourself.
तुम्हाला काय कार्यक्रमात थांबायला सांगितलंय कोणी? हा कंपल्सरी. आता एक सोपं झालंय सर, व्हॉट्सअपवर नोटीस टाकली की अप्लाय टू आलो. काय टाकले नोटीस व्हॉट्सअपवर मला? 10:30 वाजता हजर राहा. असं नसतं काय? पण ते व्हॉट्सअप नसेल तर मग काय करणार ते? व्हॉट्सअपवर कंपल्सरी आहे का आपल्याला? काय नाही, व्हॉट्सअपवर टाकणं तर तिथंच आहे ना. तुम्ही मोबाईल का
Can you suggest what we have to do now?
एक FDP चालू आहे. आवाज तिकडे
Can you
Thank you,
tell me
sir.
what we have to do?
Thank you, sir, for equipping us with the recent advances in anion sensors and its different approaches, calorimetry sensors and cyanide sensing in food machines. Once again, thank you, sir. Now it is time for a tea break. Participants, kindly rejoin the next session at 11:45 a.m.